All right, picking up where we left off, we kind of went through how this complex 1 actually achieved its job, but looking back at complexes 3 and 4, they work in very similar ways. Again, passing electrons onto combinations of iron sulfur clusters, copper ions, and other various groups embedded within these proteins. And all of this causes protein or protons to be pumped into this inner membrane space. Now, this is the flow of electrons based upon input from NADH. But if you recall, there was another spot where we gained electrons, and that was transferred onto FAD. And that is occurring right down here from succinate to fumarate, right? So here we're transferring those electrons off of one of these protons, resulting in a double bond, and this is going on to FAD. Now, if you recall, while NAD is typically soluble and is bound and released from proteins, FAD is typically a fixed part of a protein system. It doesn't float off or move. It always stays put. So the question is well, what becomes of those electrons? Just so happens that the enzyme that carries out this process is actually embedded in this inner membrane, right? So it's right alongside complex 1, 3, 4, and 5. And so this works as an alternate path for electrons in that they come in, they get transferred on the FAD, and this enzyme transfers them from FAD onto Q, which, if you remember, is membrane soluble. That will transfer those electrons back to complex 3. So in reality, there are a couple of ways to get into this complex, or to get into the system. Most of these are going through complexes that transfer protons from one side of the membrane to the other, and these are all going to end up running back through complex 5. So here we have in red flow of electrons. You can see that all those electrons are passing through these systems, ending up onto water. And protons are pumped by complexes 1, 3, and 4, all supplying protons to run back through this proton water wheel of sorts. Now, our learning objective here was to compare the mechanisms of ATP formation during glycolysis and oxidative phosphorylation. What we've given you is half of this oxidative phosphorylation. Now, of course, we've already seen what happens in glycolysis. That is all coming from substrate level phosphorylation. And what we've shown you here is kind of how oxidative phosphorylation works, but there's an added component. That added component is understanding chemoosmotic coupling. Okay, so there's kind of a paired learning objective that works with this. So if you recall, we are pumping protons from complex 1, 3, 4, and all those protons are running into this inner membrane space. They're then going to run back through this complex 5, which generates ATP. Something to think about with the protons in this inner membrane space is this is creating both a chemical gradient in that we have hydrogen atoms over here that are in higher concentration than over here, but there's added components to this too. So all these protons have a positive charge. That is going to leave an electrical gradient as well. So we have positive charges in this intermembrane space, a cluster of negative charges in the matrix. In addition to that, we have a pH differential. So this intermembrane space is going to be more acidic than the matrix itself. All of these factors are working together to create a combined electrochemical gradient. So there's both an electrical component and a chemical component to this gradient, both of which are driving these protons to want to cross this membrane. So then the question becomes, how do the protons get there? And that, of course, is through this complex 5. This is also known as ATP synthase. And it is, again, a multi-protein complex that has several parts, and they are moving parts. Protons will pass through a specific channel here, and it will turn this F0 complex. So this F0 actually spins within the membrane, and it has a kind of a post that sticks down in the middle of this F1 complex. This F1 complex has proteins that change conformation as this rotor spins, and the stalk complex here is holding the F1 still. So F0 is spinning, F1 is held stationary. Here is another look at how this works, so we get an idea of more of the actual procedure, or how the protons flow. So here the proton comes in, there's a little hole that runs into this slot in between two of the subunits. In between each of these subunits, there is a proton binding spot. So the proton comes in there, binds, and there is one spot where the protons line up with a hole that allows it out the other side. Notice that this is one full rotation away from where the protons come in. So as this one leaves, this whole thing moves over one, a new proton comes in. This proton in this space then goes out. It turns one more partial turn. And as this rotor turns, it's turning this internal rod, which is moving an asymmetrical unit inside of this catalytic knob. And this is changing the conformation of the proteins in this catalytic knob. Now, this is very similar to a water wheel, right? So we have this head race up here, the water is pouring into these slots. Gravity is then forcing rotation in this wheel, and then they come out the other side. It is the exact same mechanism, okay? Now, if we take a look at the post that sticks down through the middle of this complex right here, this catalytic knob. So here is the catalytic knob. Here is that internal post. Notice that it is asymmetrical. Okay. The catalytic knob, it's made up of alpha and beta subunits. So here's one pair, here's another pair, and here's another pair. The beta subunit is actually responsible for ATP formation. Notice here that this knob running down, or this rod running down through the middle, is asymmetric, and as it makes contact with each one of these three different beta subunits, it causes a different conformation form. So when you have this little post thing, it is in this form. When you have this little notch, it's in this form. When it's this little flat side, it's in this form. So here we have the open form. This is, as the name suggests, open, allows ATP and phosphate to bind, or allows completed ATP to release. What happens is when you get rotation of this, it causes each subunit to shift conformation. So this open conformation will now shift to a loose conformation. A loose conformation holds ATP in inorganic phosphate, and doesn't allow them to leave. The third possible conformation is this tight conformation, where we actually take ADP, an inorganic phosphate, and squeeze it together until it bonds. Okay, so it's really physical force holding those two things together. There is some catalytic amino acids as well, but you get the idea. Okay, so as an example, we can see how this looks in rotation. And you can see how dramatically these subunits change conformation. Just again, real quick. So this ATP synthase is a really complex piece of physical machinery that is allowing the formation of ATP. Now, always remember that we have an electron donor, we have an electron transport system that is embedded in a membrane, and we have a terminal electron acceptor. Our donor is food, and that is a source of high energy electrons. We are going to dump those low energy electrons that spend their energy in this electron transport system, dump them onto oxygen and make water. The electron transport system is responsible for pumping protons from one side of the membrane to the other side of the membrane, creating a proton motive force or an electrochemical gradient. And again, we can use this gradient to do work such as make ATP, like we've just seen with ATP synthase. Okay. Now, understand that this isn't the only thing that these protons can do. And this explains why we say it's approximately 32 ATP that is produced per individual glucose molecule. Okay? The reason why we can't give you an exact number is this proton motive force drives other things. Okay? So you have to get stuff into the interior of the mitochondria. Most of these transporters, instead of burning ATP to transport things from one side of the membrane to the other, just simply use proton motive force to co-transport stuff in as it allows these out. Okay. Of course, this in bacteria can also do mechanical work such as swimming. So in bacteria, there is another mechanical motor, kind of like ATP synthase, except for instead of turning a wheel which generates ATP, it spins a motor attached to a flagella, which works kind of like a propeller. And again, in bacteria, we don't have to only use oxygen. We usually have alternative terminal electron acceptors. And in plants and some bacteria, we can actually take electrons off of water, kind of the exact opposite of what's happening over here, and then use light energy to energize them, and then use that energy to reverse this process making food. Bacteria can sometimes also glean those high energy electrons directly off of inorganic substrates. So now we have the full compare the mechanisms of ATP formation during glycolysis and oxidative phosphorylation. We've closed this loop here, and we've also talked about what a electrochemical gradient is and what it does for us.